Thank you very much for giving me the opportunity again to talk about our work. And after the meetings we had in Portugal, where I presented my work in Cascais, I remember it was a very, very nice meeting, and I'm still very keeping very nice memories of that. And I'm grateful for the organizer to organize now the webinar series on sphingolipids. So briefly, I'm working at CNRS in France, in Bordeaux. And I'm mostly working on lipids and trafficking of polarized proteins in plants. So briefly, I did my PhD in Paris, and then I went to Sweden for a postdoc for almost 7 years before I came back to France as a group leader in 2012, where I started more or less my group in 2016, actually. So, and today I will present a novel mechanism we discovered in the sphingolipid-mediated phosphoinositide homeostasis trans-Golgi network. So, but let me first introduce the developmental background of our work and the physiological aspects. So as I said before, we are working on plants, and one feature of plants is their adaptability and growth flexibility in response to environmental changes. And I mean, we have a very good example, which is phototropism that was described more than a century ago by Darwin, where he described that the plant is able to bend towards the source of light, for example. And if the source of light is changing the direction, then the directionality of growth will change as well. This happens as well in the roots, but in some species, the root try to avoid the light. We call that root light avoidance. If you change the gravity vector, for example, like turning the plate where the seedlings on the Arabidopsis plants are growing, and then you will see that the root is bending towards the new gravity vector. So those bendings described, they are all based on auxin concentration. So auxin is a phytohormone. It's an hormone of plants, and auxin is able to concentrate at some zone of the tissue of the plant, and the concentration of auxin will modulate the growth and the directionality of growth. So auxin is a very small molecule which is able to be transported from cell to cell, and is able to be concentrated at certain places. So as I said, what is making the transport is actually the auxin carriers. And the auxin carriers, they are localized polarly into the cell. Like if you take the example of pin 2, for example, pin 2 here on the left upper side of the slide is an auxin efflux carrier. So it means that it's able to efflux the auxin from the cell to the apoplast. So pin 2, this auxin efflux carrier, is localized at the apical membrane only of the cell, of epidermal cells. So this is a very striking example of cell polarity in plants. Pin 1, which is another auxin carrier, is localized only at basal membrane of the cell, for example. Then we have other auxin carriers that are localized either at outer lateral domain or other carriers that are not auxin carriers, but other carriers that are localized at inner lateral domain of the cells. So cell polarity in plant is very complex because you can see that we have at least 4 polar domains that can be distinguished, and those polar domains establishment and maintenance are at the basis of distribution of auxin. So it's very important to understand how this works, basically, how this is working, and this relates to endomembrane trafficking. So all the auxin carriers which are localized at polarly, I mean at the plasma membrane, at some point will cross the endomembrane network. And one very important station at the endomembrane network is the trans-Golgi, what we call trans-Golgi network. So the trans-Golgi network is it's a membrane compartment actually at the transmost side of the Golgi apparatus. I will talk to that a bit more. And this trans-Golgi network in plant is able to receive the secretion, but as well the endocytosis. So it has been shown through different studies that PIN2, the auxin carriers I've been showing you before, which is localized at apical membrane of the plasma membrane, is able to slowly diffuse. Also very slow, this diffusion at some point will label all the cell, and some mechanism, some localized endocytosis mechanism are able to take the slow diffusion of PIN2 at the side to basically traffic it to the TGN and to sort it again at apical domain in a polar fashion. So it's what we call the polar recycling, and the secretion is also polar, but we don't understand the mechanism yet. So, and this is on what we are working now. So the trans-Golgi network in plants is a bit different from what we see in animals. Basically, in animals, the trans-Golgi network is attached to the Golgi and is in contact with the endoplasmic reticulum. As well, there are some exchanges between TGN, the trans-Golgi network, and some endosome, either recycling or early endosome. In plants, the TGN can detach from the Golgi apparatus and become completely independent. So the dynamic can be, of course, very different from what we found in animal cells. And this TGN has been shown in plants to be an early endosome and a recycling endosome, actually. So it fulfills different functions. So we know from the animal field, of course, that the trans-Golgi network is a very important station into sorting proteins to different polar domains, as it was shown in epithelial cells, which are polarized. We know as well that sphingolipids are enriched at trans-Golgi network in animal cells, as it was shown here for ganglioside and globoside. And we know that the trans-Golgi network contact site with endoplasmic reticulum, it's a place where you have lipid exchanges between sphingolipids in the form of ceramide, which is like transfer from ER to trans-Golgi network. And then glycosylation of this ceramide will actually activate other pathways on the homeostatic path of the phosphoinositides. And phosphoinositides are very important for the sorting of proteins at TGN, so it's important to keep this concentration. And we know that the transfer exchange of PI4P and cholesterol is mediated by sphingolipid fluxes. So in plants, we took an approach to characterize the lipid composition of TGN, and it's what we did by immunopurification, where we immunopurified the TGN using specific baits that localized specifically at TGN. So we immunopurify it, and as you can see here, we pull down the TGN as compared to the Golgi fraction here. And here it's all the Western blot. I won't details because it's quite detailed. So, but what we've seen is by characterizing lipid composition is that the TGN of plants is enriched for alpha hydroxylated fatty acids that are very long in chain. So alpha hydroxylation, as in yeast, is specific signature for sphingolipids. So we know that TGN is enriched in sphingolipids and as well sterols here, as compared to the Golgi apparatus. So we wanted to know a bit more about what is the role of the acid chain length of sphingolipids of plants. And when we used a tool which is called metazachlor, so it's a pharmacological tool which will inhibit the elongase complex. You know, the elongase complex in Arabidopsis is able to add 2 carbon at a time for each cycle to elongate the acyl chain of sphingolipids. I mean, sphingolipids, but also other lipids. And using metazachlor, you will inhibit this, and this will result in shorter chain for sphingolipids, because we have specific action of metazachlor on alpha hydroxylated lipids that are specific in nature for sphingolipids, as you can see on those graphs. So basically what we do is that When using metazachlor, we decrease the very long chain fatty acid that are alpha hydroxylated. So we shorten the chain from 24 atoms of carbons to 22 and 20. So without altering the total quantity of sphingolipids, and we don't alter the total quantity of sterols. So what we've seen is that we alter the localization of PIN2, these auxin carriers that I've presented you before, which is localized at apical membrane of plant roots. And what happens is that when we shorten the acyl chain length of sphingolipids, we lose the polarity of PIN2, and we stack it into endomembrane compartments that we identified by colocalization as being the trans-Golgi network. And this trans-Golgi network, as I've shown you before, where we see this enrichment of sphingolipids. So, as well, what we could see is that when we shorten the chain of sphingolipid, instead of having tubular connection between TGN vesicles here and the wild type control, when we treat with metazachlor, we basically prevent this formation of tubules. So the tubules completely disappear, and we have a more swollen secretory vesicles compartment, so more swollen TGN, trans-Golgi network. So this is fitting actually very well with some findings from Rihanna, where she could show that when you apply to membrane reconstitution ceramide with very long chain fatty acid in C24, oppositely to ceramide with C18 fatty acids, then you could see that tubules are forming instead of being completely spherical and round. So basically what we show is a bit similar in vivo. So, and what we see upon metazachlor using our drug that shortened the acyl chain length of sphingolipids is reminiscent with a phenotype that we see for the PI4 kinase, which is a kinase that will phosphorylate the phosphatidine-inositol, PI, to give the phosphoinositide PI4P, to produce PI4P. This PI4 kinase is localized at TGN in plants, and actually you can see that when you mutate that, you have the almost a similar effect as we can see with metazachlor. So maybe suggesting that the two are acting the same pathway. So basically we are asking the question on whether metazachlor or using the shortening of acyl chain length of sphingolipids has something to do with phosphoinositides at TGN. So, and for that, we use biosensors developed in Yvan Jaillet's group in ENS of Lyon, where he developed a full set of tools to visualize specifically different species of phosphoinositides. For example, using those tools, he could discover that PI4P is mainly localized at plasma membrane, but as well at TGN, at trans-Golgi network, while PI3P, for example, is mainly localized at late endosome multivesicular bodies. So there is a gradient of PI4P, mostly at plasma membrane in plants, but as well at TGN. So what we did is that we analyzed the PI4P localization upon the drug that shortened the acyl chain length of sphingolipid. And here we used a biosensor, which is called FAP, for the pH domain of the FAP1 protein that we introduced in plants combined with the MC3 here. So, and what we could see is that when we use 2 times this domain, or when we use 3 times this domain, which actually increase the avidity of the probe for the PI4P, we see that upon a normal condition here, on the left panel, there is no drug. You can see that we can see mostly the plasma membrane, but also some dots. Maybe you cannot see them very well, but also some dots labeled by PI4P, especially true for the 3 times FAP as the avidity is higher. And when we apply metazachlor here on those panels here, we can see that we increase the PI4P signal at TGN. And here are the quantification. As you can see, when we increase the concentration of metazachlor, the drug, you increase as well the signal intensity of PI4P at trans-Golgi network, while the plasma membrane labeling or staining or intensity is not really altered. Then we try to understand where this PI4P pool is coming from. So is it coming from the neosynthesis or not neosynthesis, but the local synthesis of PI4P from PI through the PI4 kinase, which localize at TGN? So this is the first question we ask, because the PI4P kinase is localized at TGN, and maybe we thought that maybe we will alter the PI4 kinase through shortening the acyl chain length of sphingolipid. So what we did is that we used a drug which is called wortmannin, and that inhibits the PI4 kinase. And here it's what we see, the wortmannin treatment at 5-minute treatment, 15 and 90-minute treatments. What you see is that you don't really increase PI4P inside the cell. I mean, you rather decrease it at plasma membrane as quantified in this graph. But when you apply metazachlor now together with the wortmannin treatment, we were not able to prevent the accumulation of PI4P at trans-Golgi network. Maybe we did at 90 minutes. So it means that maybe on the long run, the PI4 kinase will be important, of course, to kind of feed the full pool of PI4P. But at the short-term treatment, like 15 minutes or so, I mean, we don't prevent the accumulation of PI4P. So it means that primarily the PI4 kinase is not really involved into the mechanism which mediate PI4P accumulation at TGN through sphingolipid. So next, of course, we asked whether this could be due to PI4P sterol exchange mechanism, because we know that this is a case in animal cells. So we reason that if it will be the case, the accumulation of PI4P at TGN will be basically coupled with a decrease of sterols at the same TGN compartment. So to tackle this question, we did immunopurification of the TGN using the same baits as we did before for the lipid quantification, and using magnetic beads, basically. We were able to pull down the whole intact TGN compartment, and we quantified the fatty acids and as well the sterols. As you can see here, the effect of our drug that shortened the acyl chain length of sphingolipids is very clear. We shorten the very long chain fatty acid in the pool of sphingolipids, but we don't alter at all the quantity of sterols, while we still have this accumulation of PI4P at TGN. So we don't think that this is due to a sterol PI4P exchange mechanism. When we look at this graph in details, because this is a sum of fatty acid in C24 or C22 or C26, when we look at the details, you can see clearly that what we alter mostly is the alpha hydroxylated fatty acids that are specific in nature for sphingolipids. But we alter also non-alpha hydroxylated fatty acid in C24, and those ones can be comprised into the sphingolipid pools, but they can be also including into the phosphatidylserine PS pool in plants, because in plants PS can have a very long chain fatty acid up to C24. So maybe it's due to PS. And as we know that also in animal cells, there are mechanisms involved into PI4P exchange through coupling with PS. Then we also wonder whether this could be due to PS-PI4P exchange. And for that, we use phosphatidylserines biosensors, also developed in UVJS group, to look at what happens. And here you see the picture. Basically, PS will label the PS biosensor will label very strongly the plasma membrane, but as well the trans-Golgi network, as shown by colocalization analysis. But upon metazachlor, here you can see that we don't alter either the intracellular accumulation of PS at TGN, nor the intensity at plasma membrane. So we don't think it's due to a PS PI4P exchange mechanism as well. So finally, to identify the mechanism on which could be involved in linking sphingolipids to PI4P accumulation, we did immunopurification and label-free proteomics. to identify the proteins that could be affected upon the metazaclor treatment. And here, what we could identify is all the enzymes that are involved into grafting the polar heads of sphingolipids. So we found them at TGN, so meaning that the local synthesis place of sphingolipids is at TGN, but we don't alter the quantity by applying metazaclor. We found as well like a lot of enzymes related to PS, like PS flipase that flip PS from the cytoplasmic leaflet to the luminal leaflet, but we don't alter the quantity as well or abundance upon metazaclor. So we found as well phosphatase of phosphoinositide, like these 4 phosphatase that will basically degrade the PI4P to give PI, but the quantity or abundance of this PI4P phosphatase at TGN is not altered by metazaclor treatment. Instead, we found different form of phosphatidinoinositol, so phosphoinositide-specific phospholipase C. And those phospholipase C were clearly decreased at TGN upon metazaclor treatment. So the abundance were clearly decreased. So those PI-PLC could be the target of the acyl chain length of sphingolipids. So to check this hypothesis, we use an inhibitor of PI-PLC, which is called U7322 for the active form, and we have as well the inactive form of the drug. And we inhibit the PI-PLC with this drug and try to see what happens to PI4P. And you could see clearly that with the inactive form, no accumulation of PI4P was observed, while for the active form of PI-PLC inhibitor, we could see clearly a PI4P accumulation at TGN here, and even more strongly when you increase the concentration. If you couple that to metazaclor treatment, which increase the PI4P at TGN, you could see that there is a non-additive effect at higher concentration, meaning that maybe the two are in the same pathway. So basically, we have we phenocopy the effect of reducing the acid chain length of sphingolipids by applying an inhibitor which will inhibit PI-PLC. Interestingly, coming back to this apical marker of cell polarity we have that transport auxin in a polar way, PIN2, we can see that when applying metazaclor, you lose polarity, you have as well accumulation at TGN, and we have exactly the same when we apply this drug that will inhibit PI-PLC, but not for the active form. You will lose the cell polarity of PIN2, and we will have as well accumulation of PIN2 at TGN. So here, labeled by the dots and quantified here. We did colocalization of this PIN2 intracellular accumulation with markers either of TGN or Golgi. And what we could see is that we have a very strong accumulation of PIN2 upon the PI-PLC drug at TGN, while we have a very lower accumulation at Golgi. So clearly it accumulates at TGN. So at the end, I mean, to kind of summarize what I've been talking about now, is that, so we have sphingolipids here that are mostly, we think, localized at the luminal side of TGN. And those sphingolipids will have a very long-chain fatty acid that might interdigitate with other lipids on the other leaflet. This interdigitation might cause might cause, I say might because I mean we don't know that, but from our experiment, what we can say for sure is that this acid chain length of sphingolipids impact the quantity of PI4P on the other side of the membrane. So for which mechanism this is happening, that will be the perspective for future studies that we want to know and definitely identify this mechanism. But what we could say for now is that reducing lipids that are sphingolipids on one side impact the accumulation of another lipids, which are phosphoinositides, very important for protein sorting on another side. And we think that this is involved in the sorting of polar protein which is an auxin carrier in that case, into the apical membrane of root cells. So at the end, I would like to acknowledge people that have been working in this subject. So Nicolas Isnay was in PhD in my group, working on that, doing all the polydynamics. And Yuko Ito is a postdoc in my group, also working on that and other subjects and finishing all of this project. And with that, I would like to thank you for your attention and open for questions. Thanks.